Good evening. Okay, no problem at all. That's fine. Yes, hello, good day to everybody. My name is Dr. Mark Miller and I am a cardiovascular research scientist here at the University of Edinburgh. I've been studying cardiovascular disease for most of my research career, and then for the last sort of 12 years I've been looking at particularly at how air pollution can affect cardiovascular disease. Okay, let me just get this here. So I'm hoping that's sharing with you now. Is that good? No, it's not there. Is that there? Okay, hold on. You can see me? Good. Oh, there we go. Okay, so as I mentioned, for the last sort of 10-12 years, we've had a program of research looking to see how air pollution can damage the heart and blood vessels. And in particular, we focus on particles, and we have a special interest in nanoparticles, like those that come from diesel exhaust engine emissions. And I'm going to show you some of the research today. To start with a little bit of background, everybody knows that air pollution is bad for them, but actually the mortality figures for air pollution really are staggering. And it's now estimated that actually several million deaths are actually due to air pollution around the world every single year. And I'm going to just highlight one particular study. This is from the Global Burdens of Disease Studies. And this large group of authors, they had the unaimed task of ranking the risk factors for all different types of disease around the world in different populations. And they ranked the top 20 risk factors for disease. And they ranked them on a scale and had an estimate of morbidity or mortality. And within this list, in the top 10 risk factors for all causes of death, both indoor and outdoor air pollution were both in the top 10 risk factors. But what's particularly interesting about this study is that it broke down the morbidity and mortality into different causes. And you'll see that where air pollution is concerned, the cause is most of this light blue colour. And this light blue colour represents cardiovascular disease. And actually, this is something that the world is beginning to become aware of now. At one point it was thought that air pollution only affected the lungs, but now over the past 20 years we have understanding that it affects around the whole body. And there's a good body of evidence that shows now that both short-term and long-term exposure to air pollution is associated with a variety of cardiovascular conditions, for example, presenting to the hospital with a myocardial infarction, a heart attack. And while people have been researching this for quite a number of years now, many questions still remain. For example, so how does air pollution cause cardiovascular disease? Which are the components of air pollution which are responsible? And if you take all the epidemiology together, the one thing that comes out is when you're talking about the cardiovascular system, all the associations are strongest for particulate matter in air pollution. And air pollution is it's far too complicated for me to do justice in 30 minutes here today, but there's lots of different ways of categorizing air pollution. And if we're looking at the particles in air pollution, people tend to do this by the size of particles. And people will categorize the particles in different sizes, and you've probably heard of PM10 and PM2.5. And these are particles that have a diameter of 10 micrometers or less, or 2.5 micrometers or less. And there is a third fraction called ultrafine particles, which I'll come back to in a second. And all these measures measure the mass of particles. So they quantify the levels of particles in the air by the weight of these particles. But if you look at the trends of air pollution in many countries, certainly many developed countries, actually what you see is over the last 60 years, the actual mass of particles in the air has decreased substantially. But what this overlooks is at the same time, there's been a dramatic increase in the number of vehicles, certainly on UK roads and actually in many countries. And with this comes an exponential increase in the number of traffic-derived nanoparticles. And nanoparticles, as everyone knows, they're the smallest of the categories of air pollution. These are particles which have a diameter of less than 100 micrometers, and they're in about the same sort of size range as viruses. And combustion-derived nanoparticles in particular will be anywhere in the region of between about sort of 3 to 100 nanometers for the nanoparticle size. And what's important about these is they're believed to be the most harmful fraction of particulate air pollution, because while they contribute very little to the mass, they have a huge reactive surface area with which to interact with cells, with which to carry chemicals into the body, and also to penetrate deep into the lungs. And if we're talking about nanoparticles in the air, the major source of nanoparticles in urban air is from vehicles, and in particular diesel exhaust particles are especially rich in nanoparticles. So this led us to a program of research between cardiologists and respiratory physicians as well as toxicologists, and we collaborated with researchers in the Netherlands and in Sweden, and they have unique exposures chambers where we can actually expose human volunteers to different air pollutants. So what we have is we have an idling diesel engine, and the majority of it just passed away and is evacuated. But at this stage here, we can take a small fraction of the exhaust, so from a diesel engine, and we can dilute it with filtered air. And the important thing is you can set it at levels of whatever you want to look at. So for example, in a highly polluted city, in a mega city such as Beijing, for example. Then after this, it passes to an exposure chamber, and we have different volunteers, different patient subgroups who will exercise, and they will be exposed to the diesel exhaust, the dilute diesel exhaust, for 1 or 2 hours. And then after this time, we can go to a clinical research facility, and we can study a wide range of different cardiovascular parameters. And this was a large body of research that's all been published previously, so I won't go into any details here, other than just trying to summarize this 10 years of research in one slide here. And we showed that just this very short period of diesel exhaust exposure at the representative levels had effects throughout the cardiovascular system. It affected the blood, it made it more liable to clot, it stopped the breakdown of blood clots. It affected the heart, it changed the rhythm of the heart, it made the heart more susceptible to damage, for example, if it had a blocked coronary blood vessel. And it also damaged blood vessels around the body. It impaired the ability of these blood vessels to relax and contract. And that's important because that's needed to control blood pressure and to control the flow of blood around the body. And the one thing that all these things have in common, these are acute effects, but all these acute effects will actually lead up to making someone more likely to develop cardiovascular disease. And there is a disease called atherosclerosis, and this is the buildup of fatty and inflammatory cell-rich plaques inside of arteries, and these plaques grow and they disturb the blood flow. They can also break open, and when they break open, they can clot off an artery, and will stop the blood flow to this region so that the downstream region becomes ischemic. And this is what happens when you have a cardiovascular event. If this happens in the heart, this will lead to a heart attack. If it happens in the brain, it will lead to a stroke. And it can happen in your peripheral limbs as well, potentially leading to loss of the limb. So atherosclerosis is something that underlines the vast majority of cardiovascular conditions. And this is something we've looked at ourselves. We turned to an animal model to do this. So we turned to a mouse model called an apolipoprotein E knockout mice. And these mice are prone to developing atherosclerosis, especially when you feed them high-fat diets. So we developed a protocol where we had these mice and we fed a period of high-fat diet. And during the later periods, we administered diesel exhaust particles specifically to the lung. So this was 8 repeated installations to the lung over this time. And we did the same in healthy animals as well. And at the end of this, we will collect the arteries and we will look at the extent of atherosclerosis, and we'll look at a range of different mechanisms to see what's underlying these effects. And again, I'll summarize this very quickly. This is one of the major arteries that's taken from the mouse. This is the aortic arch, and there's a descending aorta. And this red stain, that's where there's these atherosclerotic plaques, these lipid-rich plaques. We also took specific arteries as well and quantified the volume of these plaques by serial sectioning with histology. And which either way you look at this, if we have an animal, an ApoE animal that's prone to atherosclerosis, and they just received the drug vehicle, they have a degree of plaque, and in just a short period repeated installation of diesel exhaust particles in the lung, put a big increase in the extent of the plaques. It almost doubled the size of the plaque when you quantified it by histology. So we've shown really that both acute and chronic exposure to air pollution have marked effects throughout the cardiovascular system. They promote disease. What we've really been interested in is the biological mechanisms that could account for this. So it's perhaps easy to see how particles in the air are inhaled and they go into the lungs. But the real question really is, how do we bridge the gap from the lungs to the blood vessels? How do we cause cardiovascular disease? And actually, this is something that's we're still trying to get to grips with. At the moment, there's several different theories for how this may happen. There's 3 main theories by which once particles go into the lung, they can affect the cardiovascular system, potentially cause lung inflammation, and there's a spillover of mediators which affect the blood vessels, or that these particles may actually themselves get access to the cardiovascular system, or that they can change neural and endocrine regulation. And actually, over the last 10 years, we've found evidence for and against really all 3 of these different hypotheses. And I'm going to show you just evidence just on one of these pathways today, and that's the middle pathway, particle translocation. And to show you this, to give you an example of this, this is an electron micrograph of a lung section. So the grey tissue is the lung tissue, the white is the air spaces of the lungs, and in the pink regions that have been shaded, this is the blood vessels of the lung. And what you can notice, there's a very, very thin barrier between the air spaces of the lung and the blood vessel. And of course, this is needed for gas exchange to occur. But if particles get into the lung, and these are just drawn on here, nanoparticles would be much smaller than this, there's certainly potential by which these particles could cross over into the blood. And I'll show you some of the evidence, because this is a field that's really sort of dogged this field for a long period of time. It's something that people have always thought that happened, but actually at the moment there's really lacking the evidence in man to show that this does happen. So let me take you to the later stages of this pathway first, and this is asking the question from another way around. Let's say these particles do get access to the cardiovascular system, they get access to the blood. Do they then have the capacity to cause harm? And if we're talking about particles causing harm to cells, there's many different ways that nanoparticles can harm cells to cause them to promote cell death or apoptosis. And in particular, you'll see there's one here, oxidative stress. So this is an imbalance in the generation of oxidant species such as free radicals. And actually, oxidative stress has many different actions, and because of this sort of slightly indiscriminate nature of oxidative stress, it can account for many of the varied effects that nanoparticles and diesel exhaust particles could harm cells. So the first thing we did was we had a look just to see if our particles themselves were generating free radicals. I won't go into this in any detail other than to say we use a technique called electron paramagnetic resonance, and this is just a really sensitive technique, and we don't need to worry about the dark colour of the solutions. And the technique acts by adding a chemical called a spin trap, which stabilises free radicals long enough that you can measure them by passing a magnetic field over your sample. And this is an example of a positive control you would see. And you'll see these characteristic three-peak spectrum, which tells us it's superoxide free radical. And the height of this peak gives you an indication of the quantity of the free radicals. And we can then take filters that are either clean or filters that we have diesel exhaust particles we've collected from our clinical exposures. And again, you can see when the diesel exhaust particles are there, there's a big increase in the extent of free radical generation. I will just skip this slide, just to be quick. The other thing we can have a look at is we can have a look at how these particles actually could directly affect vascular function. So you can take isolated pieces of artery and you can mount them in a machine called a myograph, where these arteries are put between two jaws and they're kept alive in a physiological solution, and you can measure these arteries contracting and relaxing. And this is an example of a trace here where we have an artery contracting, and this is a dose-dependent relaxation to increasing amounts of a vasodilator drug. And this is some of the tissue after it's been in the myograph, where we've actually directly added diesel exhaust particles to the bath. These are the diesel exhaust particles clustering on the inner surface of arteries. These are the endothelial cells, which are crucial to the function of blood vessels. And you can start to see the diesel exhaust particles sticking to the inside of the artery, right next to the endothelial cells where they would cause the most harm. And we want to see what effect this has on how well the artery can relax. So how well the artery relaxes gives you an idea of how healthy the artery is. And you add a drug called drug acetylcholine, and this causes a dose-dependent relaxation of the artery. And this is just a standard healthy relaxation. If we do this in the presence of diesel exhaust particles, you can see there's a marked impairment of the ability of these arteries to relax. They just can't relax to the same extent they would be. And you can add drugs such as SOD, and the scavenges superoxide, scavenges free radicals, you can partly reverse this signal. So it shows that these particles, if they do get access to the blood system, they certainly have the capacity to impair vascular function through the generation of free radicals. And it doesn't necessarily need interaction with the lung or inflammatory cells to do so. So let me go back to this schematic again. So I've been talking about the middle hypothesis, and I've shown that these particles do have the capacity for damage. I'm now going to talk about the first stage of this pathway, and that's really asking the question, do these particles get access to the cardiovascular system? The in vitro assays I've just shown you, they're useful. They're useful for screening particles, screening the different constituents of particles. But actually, the main question is, do these particles get access in the first place? And this is something that's really been a problem in this field for a while. And one of the reasons for this is it's very difficult to investigate particle translocation. This is for a variety of reasons. Firstly, just trying to detect the small size of nanoparticles in tissues. We know that if particles do translocate from the lung to the blood, they do it at very low levels. And once they enter the blood, there's a large volume of blood and also many organs they can accumulate in. So very small amounts of particles which are difficult to detect in a large volume of tissue. People have tried to overcome this by adding labels to try and track the particles, but in the past some of the experiments that have been done in man have suffered due to the label itself coming off the particles. Additionally, if we're talking about... Oh, sorry, So I'm just about to show you some data from this. So when people have done this before, they've either tried to use fluorescent labels of particles and they measured the fluorescence, or the other way to do it is to use a particle that's made of something that can be measured in very low amounts by mass spectrometry. And that's some of the data I'm just about to show you. What we would like to do is we'd like to do this for carbon-based particles. So the particles that come from diesel exhaust, they're largely carbon-based, and that's something that's really tricky to do in the body because the body itself is made of carbon. So the way that we ourselves got around this was that instead of using diesel exhaust particles, we turned to a model particle called, which is gold nanoparticles. And we use these for several reasons. Firstly, that the size is very similar to the particles in diesel exhaust. Also, the particles are largely inert, and that way they're safe for use in human and animal experiments. And the point you've just raised there is the specialized techniques where you can measure these very low levels of nanoparticles. So we use a technique called inductively coupled plasma mass spectrometry. And in particular, we have collaborators that have a very high sensitivity machine that can measure the very low levels that translocate. So because we work with clinicians, we first actually started doing this in human studies. We have experience of doing exposures to diesel exhaust nanoparticles, so we changed this to looking at gold nanoparticles. And then we generated gold nanoparticles by spark generation from an electrode, and these generate very small nanoparticles in the region of about 5 nanometers primary particle size, which cluster slightly in the air. And we asked healthy subjects to breathe these gold nanoparticles for 2 hours while exercising moderately. And then once they've had the 2 hours exposure, we collected blood samples and we collected urine samples. And this is some of the data that we found. This is quite a busy graph, but this is looking at the concentrations of gold in the blood. This is the concentrations of gold in the urine, and we're looking at over time. So we have the end of the exposure, and we're looking at the next 6 hours, and then we invited people to come back at later dates. And what we could start to see, even though we're really at the threshold of detection, so again it's very, very low levels of gold in the blood, we did start to see a small spike in gold nanoparticles appearing in the blood within about 15 minutes. And over the next 6 hours, there was a trend towards an increasing levels of gold in the blood. And by 24 hours, we could measure gold in the blood of almost all of our volunteers. And what was especially surprising as well is that we had the opportunity to invite our volunteers back 3 months later, and we could still detect measurable levels of gold in the blood at that time. Also, we could measure quite high levels of gold in the urine, both the day after and 3 months later. So it does suggest that these particles are accumulating in the body and they're persisting for long periods of time, and actually excretion of these small particles in the urine is one of the clearance mechanisms. So that was our first result, and then we started to ask other questions to do more with the mechanisms. So one of the questions we wanted to ask is, how does the particle size affect the ability of particles to translocate? So for our clinical exposures, we have our primary particle size, which is around 4 to 5 nanometers primary diameter, and we also modified these particles as well to actually aggregate and fuse together particles to generate a larger particle with a primary diameter of around 30 nanometers. And we did the same inhalation exposures, and this was the results we had with the blood, where you can see that the small nanoparticles much more readily translocated into the blood than the large nanoparticles. Okay. That's right, yeah. So a lot of air pollution, and the way air pollution is conventionally measured, is by PM10 and PM2.5, and these are much, much larger particles. And the reason we focus on diesel exhaust is it's much smaller particles. So the primary particle size of diesel exhaust, it varies quite a lot because it has a quite complicated size distribution depending on the fuel and the engine. But actually, if you look at it, the primary particle diameter of diesel exhaust, it tends to be somewhere between about 10 to 30 nanometers primary diameter in the exposure studies that we've done. So that was one of our reasons for choosing to look at slightly bigger particles, is that we knew that the primary diameter of 5 nanometers was a little bit small for diesel, although there are still diesel particles of that size. But this sort of larger particle size is getting towards the upper size range of diesel exhaust particles. So our reason for doing this was to look specifically at diesel exhaust particles and to perhaps say that actually we need to consider how we are looking at air pollution, and instead of just looking at any size particle in the air, we need to specifically look for sources which are very small or have ways that could damage the bodies further. Okay, so is that okay? Okay, so we did the clinical exposures where we had a 5 and 30 nanometers, and at the same time we did a parallel experiment in mice, and this way we could purchase nanoparticles in a much bigger size range. And at this time, just as with the clinical studies, there was more of the smaller particles translocated into the blood, and this got less as the particle size increased, and there seemed to be a cutoff between about 10 and 30 nanometers into the blood. And also with the urine as well, again the smaller particles more readily got into the urine. This time there was a threshold effect somewhere between about 5 and 10 nanometers, and this fits with the sort of pore sizes that the kidney has. The one thing we do note with all of this is that we are looking at very, very small amounts of particles, very small numbers of particles. So actually just showing these things getting to the blood is not necessarily important. What's perhaps more important is where they go once they are in the blood. And we want to ask the question is, do they go to areas where they could cause most harm? And we know that nanoparticles associate with inflammation, and inflammation is a major driver of vascular disease. So we turn back to our model of the vascular disease atherosclerosis, the mouse model, and the mouse model is nice because different parts of the vasculature get different levels of disease. So there's a region here called the aortic arch region, which gets very inflamed, where there's another large straight artery which gets very little disease. So we administered gold nanoparticles to the mice, to the lungs of these mice, and then we, the next day, we sacrificed the mouse and took out the arteries, and we separated out the different arteries. And what we could see is that in untreated animals there was very little gold or no gold, as you would expect, but we can actually start to measure gold nanoparticles accumulating in the vascular tissue. So not just getting to the blood, but accumulating into vascular tissue. And probably the most important result we saw was the levels of gold were much higher in the vascular tissue with disease than the vascular tissue with a lot less disease. So it sort of suggests that these particles, not only do they get into the blood, but they're preferentially accumulating at areas of disease. And this was in mice, and we did one final, first we were trying to visualize these particles. This has been something that's been quite a challenge. If you look at the lung, you can see macrophages that are heavily laden with gold nanoparticles. Looking in the vascular tissue or in the vascular disease is much more difficult. We did occasionally see clusters of suspicious particles, which could be particle aggregates, but we have difficulty at the moment saying that these are actually definitely translocated particles. The one thing we did do was do some Raman spectroscopy, and we stained these gold particles with a silver stain, and this allows you to have a characteristic spectrum. We then took the vascular tissue, either the lung or the atherosclerotic plaque from these animals, and again we could start to see this characteristic signal. So it does suggest that these gold nanoparticles are definitely getting to the area of disease. And the Raman spectroscopy is important because it's specifically looking at particulate gold, not at soluble gold. And we did one final experiment, and that was we wanted to prove that the same sort of thing happened in man. So we had the opportunity. This is in human, yes. So we had the opportunity in human, where we do lots of clinical studies in our hospital, and we have people who are having surgery to take out the disease in the major artery of the neck. So the carotid artery that goes to the brain. We have patients who have previously had a stroke, and one of the reasons for a stroke could be that the major artery in the neck is blocked off, and there's a surgical procedure that people have where the actual major artery is opened up, and actually the area of disease is taken out, and the artery is then sutured. And this is shown to be beneficial. It sort of prevents the likelihood of further strokes in these particular patients. So we managed to recruit a small number of patients who were happy to actually have an inhalation exposure of gold nanoparticles the day prior to the surgery. And it was a small number of people. This is just an example of some of the surgery. It was a small number of people, but afterwards they consented to giving the piece of diseased tissue that came out of the artery afterwards, and we could section this and we could scan it with Raman microscopy. And again, we saw this characteristic gold spectrum that is distinctive of gold. And this is a region of vascular tissue from someone who hasn't received the gold exposure, and this was the region of vascular tissue from someone who had the gold exposure, and you can see this distinctive cluster that is an intense signal that is characteristic of gold. So that just leaves me just to quickly conclude. So I just recap to say that air pollution, it's a serious issue. It's a serious issue around the world, particularly in relation to cardiovascular disease. And diesel exhaust particles in particular are important because they impair cardiovascular function by many mechanisms, many that I've not had time to go through today. One of our important findings was that inhaled nanoparticles can actually translocate from the lung to the blood, and they preferentially accumulate at sites of vascular disease. And really, the next question we want to ask is, do combustion-derived nanoparticles, do environmental nanoparticles behave the same way? Because if these particles do also reach areas of the vasculature which are susceptible, they're very likely to promote disease and potentially trigger a cardiovascular event like stroke or heart attack. And our overall public health implications really are that pollution, it needs to be tackled. It needs to be tackled urgently, and reducing emissions from vehicles, particularly vehicles which generate large numbers of nanoparticles in their exhaust, would be a way of potentially reducing the cardiovascular effects of air pollution. And of course, I should just quickly acknowledge all the many people who are involved in this work. It was a large program of work with many collaborators, and we also have different funding sources. I'd like to particularly highlight the British Heart Foundation, which funded the vast majority of this work. And I'm happy to take any questions. If you
Thank you.
There we go. Okay. Well, there's a lot of research in there. So when we first did our research to do with diesel exhaust particles, we had consecutive funding from program grants by the British Heart Foundation, and that was, I think, in the region of several million UK pounds to do the air pollution work. To do the gold experiments, it's not necessarily the cost that's difficult. The logistics are challenging. So we need to bring together lots of different people. So this work involved collaborating with the institute in the Netherlands called the RIVM, and they have very specialised exposure chambers and specialised expertise and technicians, and they bring their facilities across to the hospital in Edinburgh. And that that has a cost to it, and it's quite logistically challenging. And at the same time, we then need to coordinate both with scientists like myself, with cardiologists like myself. We have to try and find the availability of clinical research facilities, and of course have to factor in the availability of the volunteers. So really the logistics have been something that has been challenging. So the gold nanoparticle work, I think, has taken in the region of about 5 to 6 years to generate that body of data. So we're pleased to see it finally out and published now. It would be as well, £1 million UK would be about $1,200,000 United States dollars. Sorry, could you ask that again, please? Oh, we've had a large body of researchers. So at any time, we had in the region of about 10 researchers working at once over 5 years. And you can see from my acknowledgement slide, there was lots of different collaborators involved. The main thing that made the work so successful was having people from different backgrounds to do the work. So we had respiratory physicians and cardiologists with clinical experience. We had excellent collaborators who had great expertise in the exposure on work, and we worked with a lot of specialist toxicologists who did a lot of work into nanoparticle toxicology. So we had a big team of different backgrounds, and actually it's working together like this has generated the most interesting results. That's right. That's what I think we'd really like to see now. So while our background particularly is in a lot of the biological mechanisms, trying to understand the biology of this, but really we're keen that the work does not get overlooked by policymakers. I think it's really striking. Everyone is aware that air pollution is bad, but it's really striking just how damaging it is in the body. And with our diesel work, we noticed that really just very short period exposures had really dramatic effects around the body. And actually recently, if you look at other people who work in the field on different organ systems, it has effects throughout the whole body. People are starting to show it has air pollution has effects on the brain, on the gut, on the reproductive system, on stem cells. It really affects everywhere. And so this can't be taken lightly now. And I think this does need to be translated to policy. So we are trying to get governments and certainly various different agencies to recognise there is a problem and to put pressure on it so that policies do change quickly. That's right, yeah. Some of the estimates are really staggering. I don't have the figures just now. They tend to vary a lot, but most of the figures tend to be in billions of US dollars due to air pollution. But it's very hard to estimate this because a lot of the effects of air pollution will not just be from people presenting at hospitals, they will also be due to general ill health. And there will be a lot of people who potentially could have their lives shortened by air pollution, and that's not necessarily picked up by the doctors. And that's hard to quantify. This could affect people's working lives. So I think really that if you're looking at economic costs, they really are staggering. That's right. That's right, and I don't think Sao Paulo's alone there. I think most cities are actually increasing the number of cars. I think there's only a very few cities that are starting to decrease them. I mean, cars are very popular. And even cities around the world that have a bad reputation for air pollution, so I'm thinking some of the Indian and Chinese cities, where there is a real concern about this, and the government itself is really trying to try and control air pollution, there is still an increasing uptake in number of vehicles being produced. It has to be said that a lot of the modern vehicles are a lot cleaner than they used to be. The modern engines are a lot cleaner, and there's lots of now alternative fuel sources, but they tend to be the most modern cars, and there's still a large history of older cars which are heavily polluting still on the roads. Controlling the fuel is one way to do it. I think there are ways you can increase the efficiency from changing the fuel. You can also add additives to the fuel. So we have some research that we've done where people have added cerium oxide nanoparticle additives to the fuel, and that increases the efficiency of burning. And that sort of helps lower the amount of particles that comes out of the exhaust of the vehicle overall. I think all these things help, but ultimately we are looking at moving away from fossil fuels being burnt, because there will inevitably be some type of emission coming from fossil fuels. And I think it's really the transition to alternative fuel sources like electric vehicles or hydrogen vehicles really have to be the way forward. They have to have the infrastructure to change these vehicles that have either no or negligible emissions. It is difficult because these things do have large cost implications, and especially for countries where they have to have lots of other things they will have to spend their money on, it is difficult. So modern vehicles are expensive. Electric vehicles are particularly expensive. Changing an entire bus fleet will be incredibly expensive. But I think that needs to be the aim, and that needs to be the sort of long-term aim. And I think that people need to work together in doing that in a way that can try and finance itself to try and make these sort of changes happen fast. I think that would be difficult. I think it would be almost impossible to do it case by case. I think people have to do the experiments where they can look at some generalize, if people can get to the bottom, for example, of which physical chemical features of a particle are the most important, I think that would be the ultimate aim, because undoubtedly the composition of air pollution, the different types of particles that people are exposed to and how they behave in the body is something that is very different for everybody, depending on how they're exposed, what sources they're exposed to, how their lifestyle changes. So I think a lot of research in this area now is just trying to work out, well, which are the most harmful features? Is it, for example, the ability of these particles to generate free radicals? Is it, for example, that particular metals in diesel exhaust particles are especially harmful? Could those be identified? Could they be removed? Is it only the smallest nanoparticles that are harmful? Do we need to find out ways to remove those? Put less of an effort into larger particles? I think those are the questions that people are trying to do. So it's really trying to find sort of general rules, common themes as to what is harmful. That I think is the only way to take that forward. I don't think people really know that completely, and it depends on the sources. So from our line of work, it's really shown that it appears to be things like diesel exhaust particles that are particularly harmful. And the reason why we think they're particularly harmful, as opposed to other types of nanoparticles from different sources, are, one, the very small size, and that small size might mean they'll get access to the blood. And secondly, the small size means they have this huge reactive surface area. And we know that things like nanoparticles from vehicle exhaust contain a huge array of chemicals, many thousand, tens of thousands of chemicals on their surface. So that these very, very small particles have the capacity to carry very many harmful chemicals deep into the body. And I think that's why we believe that things like diesel exhaust particles are the most harmful. I'm sure that in the future people will identify other sources of nanoparticles that are harmful. I know there's attention, for example, in particles from shipping or from aircraft, and these are very different chemistry, and it could be that those are more harmful. But if we're talking about general urban air pollution, and someone had to ask me which are the most harmful particles, I would say it's the particles from vehicle exhaust that have the most potential to be harmful to the body. So could you repeat that, sorry? Yes, yeah. I would agree with that. I would agree with that, and certainly I've participated in workshops where we've had experts from nanotoxicology, from manufactured nanomaterials, and also experts in air pollution research. And you are right. There's a big overlap, and there's a lot of knowledge to be gained from each side of the field. If we're talking about many of the features which we know are harmful for nanoparticles in air pollution, they tend to be a lot of the same features crop up in nanoparticles that are manufactured that could be harmful. And in that, I'm thinking of the high surface area, their ability to generate free radicals in particular. I think that's a really important feature. The complexity of particles in general air pollution is more of a challenge, but I don't think that means there can't be lessons learned between the two fields. Yeah. I think the field certainly does need more funding. There are one of the main issues they have in both of the fields, in both of air pollution research and in manufactured nanoparticles, is that there's such a wide range of different nanoparticles with different characteristics. So when people are manufacturing nanoparticles, there are many more surface modifications, many more particles made of different substances. The same is also true in air pollution as well, when the composition of these different air pollutants is very varied. And actually we need a way that we can very quickly screen nanoparticles. So people are trying to develop simple high throughput in vitro assays where they can test lots of different nanoparticles and predict their in vivo toxicology. And at the moment there's some good assays out there which give you an idea of how likely these particles are to be toxic, but I don't think we're there yet. I think there's still a need to do in vivo models and human exposures, and I think funding in the future, a lot of funding will actually have to put on developing better in vivo models, better in vitro models, like cell culture models, finding the better assays, finding the molecules which are particularly important, in the hope that we can screen different nanoparticles quicker. Not. Mhm. Yeah. Yes, I'm happy to do that. Happy to do that, and thank you very much for the invitation to give the presentation. I'd be delighted to do that. Yes, that's very good. Well, thank you very much. Thank you.